Acetylsalicylic acid is a drug that you must have taken or at least heard about. We live in a world where thousands of drugs are available. For this to happen, scientists must elucidate the path that the drug takes in our body, as well as how it interacts with our receptors. Salicylic acid, the hydrolyzed and active form of the drug, is a ligand that interacts with the protein in our cells. But if this happens too fast, too slow, or in the wrong place, it may have no effect or even produce side effects. One of the most common and easy ways to study this interaction is with the equilibrium dialysis method.
In our project, we first found the correct wavelength where salicylic acid concentration should be measured in physiological conditions with a phosphate buffer. We also constructed a calibration curve using 5 different dilutions with set conditions that will help us measure the changing concentration with time in our equilibrium dialysis with a linear equation. We proceeded to our final goal, to make a protein binding study. For this, we used pork gut and egg membranes to separate the protein and ligand, only letting through the ligand to bind with the protein. We must measure the different concentrations of salicylic acid every 5 minutes for an hour. This would tell us how much of the salicylic acid bound to the protein. This part of the experiment was pretty time consuming and honestly quite boring. After measuring the samples, we found that we did not have the reproducible data in our results. The membranes did not block the proteins from going from one side to the other and interfered with the lecture in the spectrophotometer, giving us a concentration above the limits of the wavelength. When we looked at the global data, we did not find a pattern in how the concentration was changing, and there was no mathematical model that adjusted to our data. It also didn't resemble the polynomial model of other protein studies.
For future studies, we recommend not to use pork gut membrane and be very careful with the egg membrane so it doesn't get damaged. Also, using a color reactant like tinder will help measure the concentration in a wavelength far from the wavelength in which proteins are studied, and will take out this error source.